of you who don't know me, I am actually a medical doctor trained in genetics, and I take care of individuals with CDG, but I'm not a gene therapy expert, although I would be the doctor that would be prescribing gene therapy if it became available. And I would be involved with clinical trials when it comes to that. So I'm going to talk about this topic in that perspective. And what I want to do today is to discuss why CDGs are genetic disorders and why genetics is relevant to the current and future care of your family. So to do that, I'm going to give a little Genetics 101 so we're all on the same page, and then talk about why genetics is a group, CDG is a group of genetic conditions, and then dive into the era of gene therapy and touch on that. So a little history about human genetics and CDG. The concept of hereditary, that traits can be passed on from one generation to the next, has been around forever, since ancient times. Pythagorean was talking about it. But it wasn't until the early 1900s, like 1901, where the word and term gene was actually coined. And it wasn't until the 1930s that genes were found to be made out of this chemical called DNA. And the 1950s was when Watson Crick and Franklin Roslin discovered the structure of DNA. And then following that in the 60s, it was discovered how DNA was read and translated into proteins, which is like the workhorse of the human body. And then it was in the 80s when the first CDG was described by Professor Yakin. And then the genes that led to the CDGs were first mapped in the 80s and 90s. And then over the last couple of decades, the number of genes that are misspelled that lead to CDGs have exploded. And a few of the people who contribute to this explosion are sitting in the audience. And we just started a new decade, the 20s again. And is this history, time in genetic history about gene therapy? I think it will be definitely a part of the 20s and 30s. So Genetics 101, from all of the science and a few Nobel Prizes, we've learned that the human body is made up of about 3 trillion cells. And every cell, almost every cell, has a full complement of DNA that's kind of housed in this part of the cell called the nucleus. And the DNA is packaged into these structures called chromosomes. And the chromosome is really just a very tightly wound, very long chain of DNA. And what the DNA is, it's really actually a series of nucleotides, these are chemicals, that we just name ATCG for short, so that we can kind of talk about it. And it's kind of the order of the ATCs and Gs in this very, very long strand of DNA that's attached by a backbone that determines what the protein is made. And to kind of look at that, how DNA is translated into protein, is actually called the central dogma of biology. So you have DNA, which is kind of the storage of how we have our genetic material stored. And in order for that to be able to be translated into a protein, it first has to actually be transcribed into RNA. And that RNA has to actually be processed by a process called splicing, where the parts of the RNA that encode protein, called exons, have to be cut away from the parts that don't encode protein and then attached together, and only those parts can be then translated into the protein. And every 3 nucleotides on your RNA sequence really specify which protein, which amino acid is on the chain. And so the order of the nucleotides on your DNA and RNA specify the order of amino acids in your protein. And it's really the order of the amino acids on your protein that determines how the protein, which is kind of the workhorse of your body, is folded and structured, and how it interacts with its environment and how it goes places. And so it's really the order of the amino acids, which is dictated by the order of the nucleotides, that, that tells you how to make this protein that allows us to function. And so, when you have a misspelling, so one of these nucleotides or a few of these are not what they're supposed to be, when you have a misspelling of the genetic code of the DNA, then you will have a miscreation of the protein. And sometimes this abnormal protein product can lead to symptoms or a disease. And so if the disease is due to a misspelling in DNA, we term that a genetic condition. And there are different types of misspellings that you can have. It can be what we call a missense mutation, where basically it's usually one letter, one nucleotide, that is changed for a different one, and that causes one amino acid to be changed for a different one. Or it could be a nonsense, where again it's just one nucleotide or one letter that's different, but instead of changing one amino acid for another, it makes it so that there's a stop early and you just stop making amino acids after that place. So that's usually pretty abnormal. Or you can have insertions, so you just have extra DNA letters inserted in your code book that makes things not make sense. Or you can have deletions where there's parts that are missing. Or you can have what's called a Frameshift variant, where it's usually actually a small deletion, where, as you know, each 3-letter codes for a specific amino acid. If you're deleting, say, just one, then all of the letters kind of get shifted when you're reading, and then you end up at the place you have the frame shift, having abnormal amino acids at that place and further along. And usually that even leads to stopping making the protein because there's a stop signal. There are 3 stop words that say stop in the DNA sequence early, or that it could go on much longer than it should. And so usually these frameshift variants are pretty abnormal. Or it could be a splice site variant. So what that is, again, we talked about how RNA has to be processed before it can be translated into protein. So in order for that to happen, there are actually DNA sequences within the introns that tell the spliceosome in the nucleus. This is just a thing, like a machine that cuts RNA in the nucleus to cut out the introns. And so if you have a misspelling in that sequence that tells you where to cut, then you might end up having exons that are missing. And then that's also an abnormal DNA product. And so now that we understand what a genetic disease is, it's basically you have a misspelled chapter of a book, misspelled gene leading to abnormal protein leading to a disease. I'd like to tell you that CDGs are actually a subset of a genetic condition. And a CDG is a genetic disease, and in order for a genetic disease to be a CDG, basically what has to happen is that gene that is misspelled has to be the instruction book, or has to encode the protein that is important for this process called glycosylation. And what is glycosylation? Glycosylation is the process of modifying a biomolecule, usually like a fat or a protein, maybe an RNA, with a sugar chain. And there are a lot of different types of proteins that are needed for this process to happen properly. A lot of them are enzymes, but it could be a transporter, a channel, or a structural protein needed for the cell to be made properly. But if you have a misspelling of a gene that encodes a protein that's important for glycosylation, that's a CDG. So now, because CDGs are genetic conditions, we emphasize this even in the name of the CDG. So the proper name of a particular type of CDG is now agreed upon by the international community to be the gene name dash CDG. So actually just knowing the name of your disease will tell you which gene is involved in causing all these symptoms. And for example, CDG, what used to be called CDG1A is now called PMM2-CDG. So you know now that the name of the gene that is misspelled is PMM2. So now that we're all on the same page that CDGs are genetic diseases, what about gene therapy? It's kind of like almost a no-brainer. If the primary reason you're having symptoms is that there's a misspelling in a gene, why don't we just go in and fix the gene or take it out or add a new gene? And especially in this age where there's been some successes recently with gene therapy, and even some FDA-approved gene therapies, why don't we have gene therapies for every single genetic condition? That means every single CDG. So to kind of talk about that a little bit, I'd like to first get us all on the same page about what gene therapy is. And like I said, I'm not a gene therapy expert. I'm just a geneticist, a medical doctor. So I'll let the FDA tell us what gene therapy is and how it is done.
How do they do it? Say scientists want to add back a healthy copy of a gene to cells. They need an efficient way to do it. So scientists use vectors or vehicles to deliver a gene. The healthy gene hitches a ride in the vector. Viruses are one kind of vector. Scientists modify a virus to remove its ability to cause a disease. The modified virus can then be used to carry the healthy gene into a cell. Gene therapy can happen on cells inside the body, or can happen on cells outside, and then transplanted back in again. Genes make us who we are, and gene therapy holds great promise for a healthy future.
So the specific gene therapy type that they kind of talked about there is mostly what we call gene replacement therapy. There are also other techniques for gene therapy. One is gene editing therapy that people have talked about that is done in the lab a lot with like the CRISPR and Cas9 type vector. But that's actually pretty preliminary, and there hasn't been very many, definitely not in rare disease, any gene editing clinical trials that have ever been done because of the concerns of the risk of that. There are also other types of nucleic acid therapies that are out there that we wouldn't really consider classic gene therapy. There's a therapy called mRNA therapy and a second one called antisense oligonucleotide therapy. But these therapies I wouldn't consider like classic gene therapies. Although you're using DNA or RNA as the medicine, it's not a gene therapy because they're not incorporated long term. They don't change your genetic makeup on a more permanent basis. So mRNA therapy is not giving DNA. Instead, it's giving the RNA part. And so if you give this, you would expect that now new normal RNA to be able to make normal protein, kind of a similar concept. But RNA degrades really quickly. So it's a therapy that you would give once every few weeks by injection typically. So it's more like a protein replacement or enzyme replacement therapy, more than like a gene therapy where you give it and it's incorporated in your DNA and it becomes part of you. Whereas an oligonucleotide therapy or antisense oligonucleotide therapy is where you inject a very small piece of DNA or RNA that usually can stick to a piece of your own DNA so that there's changes to how your DNA is processed. And there's been some pretty good successes with this in terms of altering splicing. And so if you put it on, then maybe the splicing issue, because you're just covering that up, will be fixed. And so that can be helpful. But again, these will degrade and you have to give it again and again. So what about gene therapy? Again, why? So now we know what it is. Why don't we have it for every rare genetic disease? So to kind of illustrate that, I'd like to tell a couple of stories about the foray into gene therapy for a couple of genetic conditions that have done a lot of work. And the first story I'd like to talk about is cystic fibrosis. So for those of you who don't know, cystic fibrosis is a single gene disease. It's caused by a single gene, and it mainly has pulmonary or lung issues. And it's pretty severe. It can lead to very early death, recurrent infections, and respiratory failure. So the story of cystic fibrosis and gene therapy began in 1989 when CFTR was discovered as the misspelled gene that caused cystic fibrosis. And so when they discovered that, it was very exciting. People thought, we know what's the cause, so we can fix it. And so right away, people took cells of cystic fibrosis patients and put it in a petri dish and did in vitro, that means in a petri dish or a test tube, gene therapy. They stuck genes into that, and it worked. They were able to put in a normal cystic fibrosis CFTR gene, and it got uptaken into the cells, and then the cells were making normal chloride channels, which is what CFTR makes. And then they also discovered that, in fact, you only need 6 to 10% of cells to make normal CFTR protein to achieve normal chloride transport. So that's excellent. We don't even need a lot of the cells to, have this work. So this is exciting. So then 3 groups made 3 mouse models within a couple of years. And these mouse could not transport the chloride normally, just like in patients. But unfortunately, these mice did not have lung disease for some reason. Then, in the 1990s, the first hype was adenovirus vector-based gene therapy. Adenovirus does cause some human disease, but of course they made the adenovirus vector to not cause human disease. And they gave this gene therapy with CFTR and the adenovirus and had the patients inhale this, since it's the lung that is affected. And what they found was it was not effective. Instead, when they inhaled it, it caused some inflammation in the lung, and they thought maybe, it was inflammation in the lung that caused it to have too much mucus, so it wasn't taken in, and so that's why it didn't work. And then soon after, there was a patient that was getting gene therapy with the adenovirus vector-based gene therapy for a different disorder who died of cytokine storm, so a big kind of inflammatory storm. And because of that, the plug was pulled shortly thereafter for adenovirus gene therapy. And the last adenovirus gene therapy for cystic fibrosis ended in 2001. Then the next hype in terms of vectors used for cystic fibrosis gene therapy was AAV gene therapy, which is adeno-associated virus gene therapy. It looked really promising in rabbit and monkey models, but when they gave it to the humans, it didn't work. It didn't do anything. They weren't able to, actually express the protein. And then so they wanted to kind of genetically engineer it so that it would be expressed more highly just by adding the promoter and such, but they ran into a problem. So cystic fibrosis gene is actually pretty big, and adenovirus is really small. And if you want to add in promoters to make more more cystic fibrosis, more CFTR, then the DNA you need to insert is even bigger, and it wouldn't fit. And so they had to do even more genetic engineering. And then so they started turning to other types of vectors, including non-viral vectors with plasmids and lipid nanoparticles, but that only showed transient expression. They only made it shortly after it was given in the animals. And then they started thinking about using retroviral and lentiviral vectors tested in animal models. They were avoiding those initially because unlike AAV and adenovirus vectors, where you give the gene therapy and the DNA gets into your cell and in the nucleus, and it just stays separate from your own DNA, if you use retroviral, lentiviral, it gets inserted into your DNA kind of randomly, and so there's a higher risk. And so, but if it's inserted, it actually sticks around longer, and it's made more, more strongly. And so they started testing those, and then new animal models that actually reflected the lung disease were then created in the late 2000s and 2010s. And then they're testing now these new vectors with the new promoters with new preconditioning, such as maybe some immune suppression or steroids before giving the gene therapies to these new and improved animal models. And they're hoping to do clinical trials soon. So the second story I'd like to share is about spinal muscular atrophy. This is a genetic condition also, but it affects the motor neurons in the brainstem and spinal cord. And if you have this condition, these motor neurons don't work properly, and if you're severe, the motor neurons control your muscles, you're not able to move. And obviously that's really bad. You can't breathe, you can't move, and most individuals with the severe kind die before the age of 2. And so the story of SMA gene therapy started also with the discovery of the gene, and that was actually later than cystic fibrosis in 1995. But they had some lucky breaks. So in 2000, 2005, they created a mouse model, and it was faithful to the human disease. So that's excellent. And then in 2009, as we know, because it's a neurologic disease, the gene therapy has to get into the brainstem and spinal cord, which is in the protected region in the blood-brain barrier. You had to figure out how to get it through there. And they found out that AAV9 actually can get into the blood-brain barrier, and it's a tropism. It likes to actually get into the CNS, heart, liver, and lung cells. And then, so they delivered this gene therapy in the good mouse models, and they found that they needed a pretty high dose to get it into those neurons. And so then they did a high-dose gene therapy to show that it was safe in mice and monkeys. And then because the disease is so bad, it's so fatal, the FDA allowed them to do a high-risk study. So they gave high-dose AAV gene therapy, and that trial was successful in 2017. SMA was added to the Uniform Recommended Newborn Screen Panel in 2018, and it was approved as the first gene therapy for SMA by the FDA just last year. And over 100 patients have been given this gene therapy despite the $2.1 million price tag. Although there are still concerns, I would still say that this is pretty successful. There are some liver side effects that seem to be a little worse than they saw in the original studies, but I would say this is pretty successful. So currently, there are 4 FDA-approved gene therapies. It's listed in the FDA website along with cellular therapies such as cord blood. So 2 of them are for cancer. One is for the Zolgensma we just talked about for spinal muscular atrophy, and the other one is Luxturna, which is a gene therapy for a very rare single gene eye disease that is injected directly in the eye. And then if you're looking at clinical trials, if you just type in gene therapy, there are 864 studies that pop up. Not all of them are really gene therapy trials, but there are hundreds. Let's put there are hundreds. But then if you narrow it to phase 3 and 4 clinical trials, there are really only 60 trials, and actually not all of them are really gene therapy trials. And then if you kind of look at the pattern, they're actually a pattern of the ones that are getting closer to FDA approval if they're successful in their gene therapy. There's a group that were traditionally treated with bone marrow or stem cell transplant that they would use the ex vivo in the Petri dish gene therapy and give back. So it's very similar to their other treatment before. There's a group of rare genetic single gene diseases that seem to be more affecting one organ system. So there are actually 4 types of rare genetic eye diseases that have phase 3 or 4 gene therapies for, likely because of the success of Luxturna. And then there's gene therapy trials for DMD, Duchenne muscular, lipoprotein lipase that affects the liver. And then there's a couple of gene therapies for lysosomal storage disease. In this condition, we know that we only need 5% of the protein to restore normal activity. So that's kind of nice. And then, of course, complex disease they try. There's a lot more patients. So lessons learned from these stories. Things that work in the lab don't always work in people. We already knew that, though. I think accurate model organisms can be important, so that's kind of helpful. And then also, kind of a theme of this, collaboration is really better than competition. So what? We have 140 CDGs plus, but once one gene therapy is approved for one group of diseases, it's just going to lead the charge for the rest of them to come. So although we're a diverse group of conditions, of genetic therapies, we need to support each other. If it's PMM2, that's fine, because then SRD5A3 will come next, because it's very similar. So how about CDGs? Where are we at for CDGs? Unfortunately, 0 trials if you look at clinical trials.gov. But it's not that we haven't made any progress. So why? There are certain barriers that haven't been worked out, of course. I mean, a lot of the symptoms are present at birth. They're prenatal and congenital. And gene therapy tends to slow symptoms or stop symptoms. But there are definitely some symptoms that are, coming on later. It's multi-systemic. And also, there's a lot of basic science that hasn't been worked up. HUD always talks about flux of glycomes. I don't even know how he talks about it. But basically, we don't know exactly how much glycosylation is needed, where and when. And that really needs to be worked out to be able to understand what and how much gene therapy is needed, and when and how to give it. There are many genes, but we kind of talked about how that not necessarily is a stopping point for that. There are missing models, but over the last 2 days, I actually learned that many models are being developed for CDGs, and they are recapitulating a lot of the symptoms. So we are moving forward on that. And the endpoints of the clinical trials are unclear at this point, but we are also working on that with the natural history studies that we're pushing forward. So although there are barriers, we don't have clinical trials, I think we are moving forward. And in fact, there are actually some publications. There's a few CDGs that are just muscle, like the alpha-dystroglycanopathies. And in fact, they have gene therapies that have already been given to animals that look pretty successful. And then there are some antisense oligonucleotide therapies that have been developed for specific mutations, splice site mutations, in a couple of CDGs that look really successful in the Petri dish. But, we're getting there. And so what can you guys do to help? Well, it'd be nice for you guys to kind of understand the genetics so that you can talk to us, and we could all be on the same page at least. And then stay engaged to the extent that you can and help raise awareness. The more awareness there is, the more talent we can suck into this field, and the more money we can suck into this field, which is what we really need for this. And always ask your questions, voice your opinion, and push us. Push us because you guys really know what you need. You guys really know what's important, so tell us. And thank you so much to Andrea for organizing this, and the children, families, adults that are here today, as well as those that have been involved in the past and who have supported the understanding of CDGs over the years. And if you have any questions, this is my team. A shout out to Jenny, who provided me with figures because I was trying to scribble things that would not be understandable to you. My genetic counselor.